All right, so thank you all. We have a nice panel here. We'll start with some questions here from the audience. So here's a question that, wow, this is tough stuff here. So is gene editing, i.e. Crispr, a future direction for PSC research?
Possibly, but more likely to be a technology used to develop new models to study the disease in the lab. I think it's a therapy, not likely because trying to identify all the genes, correct all the genes that predispose to PSC is just not I think, feasible in any realistic time frame.
I think that's an excellent question. It's a hot topic, CRISPR technology, gene editing. I would agree with previous comment. I also think that using stem cells, we can, not to sell this area too much, oversell it, but you can do gene editing on stem cells, create better disease models, as was mentioned. And I also agree with this particular disease. If it was a single gene problem like sickle cell anemia, those are the ones that will probably first be cured with gene editing. But the problem scientifically is when you try to, if there's polygenetic influence to disease that there seems to be here in multiple cell types, you would be modifying 10, 20, 50 genes, and then a lot of problems would occur in the DNA, at least with current technology. So I agree, probably as a disease modeling point of view.
All right, next question. Would it make sense to study URSO plus vancomycin in combination as a therapy? Maybe Dr. Bowlus or Dr. Hameed.
Yeah, no, excellent question again. I personally think that is one of the, we also looking at the new medications that we're going to take, but we utilize medications which are already there and which Chris has mentioned that with the different indications and specifically looking at PSC. But using the model of these clinical trials what endpoints we're looking at it and what will be the duration of the treatment. The problem is that, the patient population is because, we have a lot of new clinical trials that coming up and it's always hard, the recruitment is always difficult with this rare diseases and which group of patients to actually get enrolled in it. That is also a big thing because, and that's where we are. But I think it has a potential. I still believe that ursodiol has some role, also vancomycin, so we have it. But the problem with all the clinical trials is that also the funding is a big thing because, these are the medications. It has to be investigator-initiated or NIH, and that is also a big thing that, doing a bigger clinical trial with about 200 patients, it would be very hard to conduct, but I think with these registries and with support, I think that is a potential future. But I would love to see what Chris thinks.
Yeah, I agree. I think that's something we haven't really discussed today, which has to do with the lack of data we have, for example, in vancomycin. It's not because there is an interest in investigating it. It's just that it's a drug that's already approved and out there, and it's a generically available drug. So there's no financial incentive for an industry partner to come along and say, oh, let's study this. So we're really at a loss and require funding through NIH perhaps, but even there, it's quite difficult. So it's unfortunate that there are these potential therapies out there, but to really study them, we just don't have the opportunity because of costs. And these trials are very difficult to run because, as you mentioned, each center can only enroll a handful of patients. If you look at the studies, it's 1 or 2 patients per center, and every center is thousands of dollars just to get up and running. So they're very costly.
Are there any potentially any individual patients where this has been tried by investigators where they prescribed with URSA and vancomycin?
Yeah.
And
So I think
so
the thing is whether a personal experience, right, whether it has been used in patients, and so we all have tried it. We had patients with ursodiol, they're already on it and using vancomycin on top of it, but in everyone you talk to, had a different experience with, and the question is what we will be looking at it, whether it's just alkaline phosphatase improvement or what improvement we are looking at it, and everyone has a different parameter. But I had patients and they are like variable success. I had more patients post-liver transplant that have tried vancomycin than pre-liver transplant patients. So it is, but again, for anything, you need to have a randomized controlled trial, and I think that was one of those things. The other example is in fatty liver disease, vitamin E is a medication that not medication, it's a supplement, and which has shown had a 50% improvement in improving the fat and inflammation. But we are looking at all these very expensive medications, but there is no funding to study because this is, a medication will be the cheapest and has some more safety. And then we're working with NIH, with our network, that whether we can study it in a much bigger, and that's where the funding thing comes up.
Yeah, the reason I was asking, because very often these case studies can also be hypothesis generating, and I guess what I'm hearing is there's no hypothesis is emerging yet on the combination.
Yeah, as far as as for what I read, that's, yeah.
I think some of these questions can be addressed to some degree with the registries, and the more patient data we collect and understand, okay, which patients have been treated with what medications, and is treatment with the medication associated with better or worse outcome, that at least give us some insights. There's always problems with that in terms of trying to factor in all the other things that can confound our results. But anyway, that's an important part, I think, of these registries we're talking about, is to get a clue if there are other medications that might be out there that have a potential.
And I think the one other thing is also, when I showed the simtuzumab study, although the study was not, but we gained a lot of knowledge about the disease itself. We had so much data, and most of these new clinical trials had patients who either already on ursodiol or they had to be, so that where some of the information we will gonna get about the ursodiol, because there are patients who are not on it and they're stratified based on the ursodiol. So I think that would be also additional information which we're gonna be getting.
All right, thank you. Next question. Using decellularized liver as a scaffold, could you cut off a portion, recellularize that portion, then transplant that portion of liver and let it regenerate to a full liver after transplanting it, like a live donor transplant?
So after the original, good question, actually, very good question. After the original papers came out, there are some scientists who are doing, I think, what the question is addressing, where they tried to decellularize in a living animal part of the organ. So I think that's kind of what we're saying. I guess you're saying that you would get the decellularized liver, put some cells in some part of the liver, put that back in and hope that it regrows. And to be honest, I think those experiments need to be worked out in general. So I don't see necessarily why it would not work. If the whole scaffold was placed in and cells were located in a certain location, it's possible they can repopulate. If only part of the scaffold was put in with some cells and you're hoping that would make the whole organ, the cells would have to secrete the matrix to build the organ. And I don't know if anyone's shown that yet. I just think these approaches are more of a possibility now than 10 years ago, and I think that's a very good question.
Okay, next question. When do you think we will see the use of stem cells in PSC?
I think the pathogenesis, the biomarkers, would have to be continued to be identified. There are scientists crazy scientists out there who will just go forward with regenerative medicine and try to rebuild things. And probably that's what I'm seeing as my role, is just to continue to work on that. But it has to be integrated with really addressing disease targets in primary sclerosing cholangitis. One of the biggest problems in regenerative medicine is you build this new tissue in a patient and the disease is still there. So it just recurs in the healthy tissues. So maybe we'd have to make disease-resistant cells, or you still have to address the primary problem. But we saw several examples there today that have been used for liver. So it's possible, and I'm giving it some more thought myself, and in which instances it might be valuable. And I've continued to talk to clinical pathologists regarding that as well.
Here's another tough one. They're all tough, perhaps. But for someone with active UC and PSC not yet progressing, what would be a good time to try FMT? And is family a good donor? Maybe Dr. Hameed or Dr. Bowlus?
I think you will be the, for your IBD experience, I think you would be a better person to. At UCSF, we are doing transplant, fecal transplant. It's mostly for Clostridium difficile patient, right now C. diff patient. And they're talking about the IBD and exploring, but we are not that much advanced in other centers who are doing more fecal transplant. So I would love to hear the experience of Dr. Tabibian about what he thinks that, but there is a role. We need to understand a little bit more about the, again, we're not there yet. That's what I would think, but, you know.
I mean, I think if the UC, in this particular instance, if the UC is so problematic that it's not responding to the multiple therapies that are now out there, biological and otherwise, then one might say, why wait? Go for it now if it can potentially help the UC, although I think the studies in UC so far have not been, I think maybe Dr. Bowlus mentioned this earlier, have not been as promising as we might hope. So that's a tough question. I'm not sure any of us have a perfect answer for it.
I would just say that I would not do it outside of a clinical trial. I don't think that should be any kind of, standard therapy. Fecal transplant for C. diff, fecal transplantation for C. diff has been, done extensively, shown to be beneficial, clearly has a place there, but I think outside of that, it should be done part of a clinical trial.
Maybe we'll take one or two more questions here.
One more thing I'll just add to the previous question that I addressed. I probably didn't address it correctly. So primary sclerosing cholangitis alone without cirrhosis is still, has to be investigated with finding pathogenesis biomarkers. However, biliary cirrhosis, I believe, is something, or extensive fibrosis, cirrhosis is something that probably regenerative medicine will push forward. So in terms of a bridging to transplantation, or perhaps eliminating transplantation for a severely affected patient, I believe that's going to happen. But I misspoke. But for early disease, we have to figure it out. Would replacing cells in the bile duct eliminate the problem, or regenerating ducts? That's something we'd have to figure out a lot more work on.
Sorry about that. All right, maybe last question here. How does the PSC Forum identify and prioritize the most urgent issues to address first?
So that's a good question as well. And we use the steering committee a lot, and when we establish a new forum, we have a lot of conversations with many stakeholders from industry, from regulator agencies, and patient groups, and we basically listen a lot, and so we have a general idea of what the gaps and needs are, and then we use the steering committee to help us prioritize, where we can start with. And each disease area will be a little bit different in terms of what the priorities might be. But we found that we have the liver forum, which addresses fatty liver disease, and in that one it was a similar situation, that you really need to start standardizing how you define things and how you name things and how you recruit patients into trials, because if you want to detect the difference between the baseline and after 12 weeks or 24 weeks or 48 weeks or 2 years, you need to be very precise about what you start with. And unless we start defining that more concretely, it's going to be difficult to then say what's the difference. And that's why, for example, starting with inclusion-exclusion criteria was one of the, it may sound like a very small topic, but it's actually very important to standardize how trials are done and really try and get at the answer, to try and get a precise enough answer at the end.
Do you have a public forum for that? Like if you're deciding is the other patients can come and comment if you started to do anything or idea?
So we have, the forum meets on a regular basis. A lot of the work is done through working groups, which meet on the phone, et cetera. And we use our patient networks to bring the information to the different patients and bring the information back. We have 2 on the steering committee, but many more patients than attend our meetings. And we're always open to additional information. The meetings are by invitation. We don't have them completely open because then logistically it becomes a little bit difficult to becomes challenging. And but we're always interested in more ideas and input from as wide a source as we can get.
All right. Well, I want to thank the speakers once again for a wonderful afternoon session, and thank you all.